Okay, this is the topic that I'm gonna talk about today is about the integrin, which can be used as a sensor, a biomechanical sensor of the microenvironment. I mean that cell can bind on the surface and can sense the environment by using of different proteins. The main one is integrin. This image that is here is from my work, and here we see that HACCP cells on the topography, and we are trying to see what's happening for the cells in terms of morphology, in terms of cell differentiation stuff, and we are going to modify this structure, and we are trying to have a different shape and different distances between the pillars to get more information about the phenomenon existing between the cells and matrix. So today I'm gonna talk about mechanical transduction signaling. So at first I should say something about the cell-cell junction and cell matrix junction, because some people are studying engineering and they don't have any idea about this biology and the basic of this principle. So here you see some three different cells which connected to each other by means of cell-cell junction. Some of them called gap junction, tight junction, and here we have adherent junction or desmosomal junction. For adherent and desmosomal junction, cells bind strongly to each other by means of catering. And for the gap junction, we have a tunnel and a gap between two cells that cells can transfer material and factors to each other. Also, if you get to the base main of the cells, you see cell bind to the extracellular matrix or the basal lamina by means of two different junctions. The main one is the hemidesmosome, and the next is focal adhesion, which is not shown here. So during last decade, I guess that biologists look at the mechanical behavior of microenvironment which affect the cell's function. I mean that from the first day of our life, we are experiencing different mechanical pressure and different type of forces, which start, for example, here, which one sperm can can go to the one egg, and here the head of the sperm pressure the eggs, and finally we have one cell. During division of the cells, you see that cell again experience different type of pressure, especially in the blastocytes. In the blastocytes, for example, here in the inner cell mass, we have embryonic stem cells, which experience different pressure from the different sites. During this step, if we remove this pressure or we apply a higher pressure or lower pressure, scientists found that there are different effects on the cell's behavior. For example, in this paper which published last year, they found that by applying a higher pressure on the embryonic stem cells, cell start differentiated to the other cell lines. It means that the mechanical behavior of a microenvironment is really important, and we should care about it. So there are different kind of studies started from the last decades, and they are trying to have a different topography, different type of structure in terms of the type of the material, because some materials or biomaterials have the stiff properties, or the other make a soft, matrix. Also, if you look at the tissue engineering scaffold and the different kind of fabric which already used for the different tissue, some of them is really rigid. For example, for the bone, we need a rigid scaffold, and for example, skin, we need a softer one. Yeah, most of people don't care about the single cell behavior and don't care about what happened for the one single cells in terms of the expression of different pathway. But actually, if we go through the one cells and look at the cell behavior, we will find that each type of topography, each type of the materials can trigger different type of signaling in the cells. So in the review paper which published last year in the Nature, they showed that integrin can affect and involve in sensing of different properties of the ECM. Integrin is the main part of cell matrix junction, which can sense the force and the biomechanical properties of the ECM, and by means of the different protein inside the cytoplasm, they bind to the actin fiber. And finally, they can inhibit or start different pathways inside the cells, which we eventually we will see this system and this modification as in the tissue. I will go through this type of proteins today. For example, in this image, you see that at first start fabrication and generation of integrin protein, which is divided. It's not a heterodimer, but when they are going toward the plasma membrane, they bind to each other to make a heterodimer. In this area, they are inactive because in the vacuole we have calcium, and in this case they are inactive. And if you look at here, you see that they are bented. But when they go toward the plasma membranes, they can move around. And finally, if there is something that they can bind them, they will activate it. It means that they will open and they bind to the other proteins inside the cytoplasm. I don't want to go to the detail of the pathway, but want to show the differences of what happened here. This integrin protein has the two super family subunits called alpha subunit or beta subunits. And here you see that one of them is the alpha and the other is the beta. In the human, it's found that 18 isoforms of alpha subunit exist, while just we have 8 different type of beta subunits. And when we have active, integrin, the two subunits should be bind to each other and they should unbend it. For example, here, one integrin can be activated by when they bind to the ECM protein, or even they can sense the chemical which exists in the environment. For example, here we have the magnesium can activate the integrin. But if you remember in the cytoplasm, we have the calcium. When we have the calcium, they are inactive, while they go to the plasma membrane and will connect to the magnesium, they will activate it. In the cytoplasm, you see that there are some adapter proteins which bind to the integrin, and finally they connected to the F-actin. The F-actin also is the cytoskeleton which involves to keep the shape of the cells, and mainly they can change the behavior of the cells in terms of gene expression. I will show you later in the next slide. But I would say that between different type of isoforms of alpha subunits or beta subunits, it has been found that alpha 5 beta 1 and alpha V beta 3 integrin can recognize the RGD motif in the ECM. So what's the RGD? The RGD is the peptide sequence which has arginine, glycine, and aspartate. And it exists on fibronectin, collagen, gelatin, or different ECM protein that cells can bind on the different domains of the RGD using the integrin. And here you see that by having this integrin, at first in the low pressure, they are inactive because there is no pressure or there is no ECM maybe around or any factor that they can activate it. So when they have a higher force, they will be activated, and in this case, we have a stable integrin on the plasma membrane. And when we have a pressure which applied on the cells, at first each of the integrin can experience high level of the force. And in that case, cell sense this situation and start a different kind of signaling. And finally, we have higher number of the integrin which can have lower amount of the force per each integrin. So if we start to pushing cells or pulling cells from the different, from the different ends, or by having different kind of microenvironments, we can trigger this integrin to be activated. By activating of the integrin, if this Persian exists, cell start to producing of the integrin very fast, and finally we have high number of the integrin that can, at the end of the day, we have the lower pressure for each integrin. So if you look at from the outside to the inside, we have ECM, integrins, adapter proteins, which called taline, vinculin, paxillin, and the other proteins. It depends on the different type of the tissue and the different type of the cells. And finally, they connected to the actin. And actin fiber bind to the nucleus, and by changing the situation and or by changing the formation of this fiber, finally we have a different type and different shape of the nucleus, which you can see here. For example, this is the one cell is in the rest situation. By applying a force, you see that we have a higher force which applied on the cells. And during this step, you see that we have higher number of the integrin which bind to the actin, and you see the different shape of the nucleus. And in this step, the nucleus sends the different type of pressure, and finally we have the other gene expression pathway. So this is the whole concept of the integrin as a biosensor. At first, we have integrin in the cytoplasm, which move and go to the plasma membrane, then connected to the ECM or the biochemical factor, they will be activated. If the pressure is low, okay, it can be stable for a while. But if the pressure exists for a long term, they again start to producing the higher number of the integrin, and finally we have the different signaling which can affect the cell migration, cell behavior, cell shape, cell how can I say, cell function. For example, if we have the stem cells and we apply pressure by different forces or different condition, you see that in some area they will differentiate it. Or if we apply a higher pressure, they will die. Or maybe if we keep them as a on a soft material, they will be as the stem cells. So I'm gonna talk about the different behavior of the material, as I mentioned earlier. For example, some people use hydrogel and the other use some kind of bioglass scaffold for the bone tissue engineering stuff. In the soft biomaterial, as I said, that we have low pressure and low forces that can sense, and here we have lower number of the integrin. So if we have stiff biomaterials, cells start to producing higher number of the integrin, and in that case we have the different type of signaling, as I said earlier. So I mean that the geometry and the properties of materials is matter. So also it has been shown that by having high ligand density in this image, it's obvious that we have a lot of integrin, which is some of them bind, or in that case, some of them is free. And if we apply the force, you see that at the first, two integrin involved, and by having the force, cell again start to producing the integrin and transfer to the plasma membrane, because they are trying to have a lower force for per each integrin. In that case, cell will be happy. Imagine that, for example, we are, how can I say, we are, for example, using our muscles, and we are in the some position that is not as a normal position. In that case, the cell are not happy and trying to behave differently. So by using the integrin, cell do the different type of signaling. In this slide, I would say that by integrin on the surface of the cells, cell can sense the stiffness of the microenvironment, and also cell can I would say can interact with the different ligand density. Also can behave by diffusible ligand density. If we have the different ligands in the soluble, cell also can make a difference in terms of migration. We have two different migration in the cells. The first one called durotaxis, which cells migrate to the material has a higher stiffness. And here you see that one cell is the leader cells and the other bind to the leader cell by means of cell-cell junction. I mentioned earlier that there are different kinds of cell-cell junction. The strongest one is the tight junction and desmophomal junction. Also, they bind by means of atom junction or gap junction. Imagine that the one cells called leader cells and go to the one direction, and the rest of the cells go toward that cells. If you remember, 2 months ago, I presented a paper that showed neurulation models in vitro. They use a micro pattern islands, which has a different diameter and they wanted to model this phenomena in the development stage. If you remember that by applying different micro patterns, cell can be differentiated to the other cell lines. In the reality, we have this phenomena in the embryonic, embryo. For example, here you see that in the neural crest, the cell going toward the one direction, and we have one type of the cells called leader cells, and the other connected to each other and going toward the dead cells. In the other sides, the mesodermal layers and epidermal layers, started to proliferate very fast, and we have the higher number of the cells. And finally, we have the different stage of development in the tissue. So I would say that, for example, if we have two cells, one cell in the two different conditions, which has the same biochemical media, but if we apply different forces on each condition, we will see the different behavior of the cells. So I would say that the mechanical pressure is really important. And if we look at the different diseases that comes from embryo and diseases that related to the cell-cell junction or cell matrix junction, especially for the cancer or metastasis that we'll explain later, we will see that in the different pressure and in the different condition, cell can, for example, proliferate very fast or differentiate it very fast. In that case, we have a higher numbers, or in the other hand, maybe we have the other type of the cells. And other examples for the collective migration of the cells is for the branching of the epithelium, which occur in the breast. And in the during puberty, we have different type of cell migration, which use integrin to go to the that root or that end of the terminal bud. For example, here we have collagen fiber or fibroblasts that aligned, and we have an optimal matrix stiffness. And here we see that cell going toward that place. But in the opposite, if we have a lower stiffness, or if we have the softer matrix, cells can go toward that area. So as I said earlier, this type of migration called durotaxis, that cells went to the stiffness material. For the cancer and metastasis also, we see this type of collective migration. Here also we have a leader cells, and the other cells from the tumors move toward that point. So for example, here we have fibroblasts. In the carcinoma, we call the carcinoma assistant fibroblasts that can fabricate ECM protein in a different way. In the normal situation, the fibroblasts secrete fibronectin and the other ECM protein, but in the cancer situation, they have this type of the ECM protein, especially the fibronectin or collagen, with a different, different type of the orientation. For example, if you look at here is the alpha V beta 3 integrin, or alpha 5 beta 1 integrin, which bind to the RGD domains of the ECM proteins. In the case of cancer invasion or tumor invasion, fibroblasts secrete this fibrinogen with the different peptides. I mean that, for example, in the normal situation, if we have the normal periodic, sequence of the peptides on fibrinogen or collagen, in case of the tumor or tumor microenvironment, we have different spaces between this peptide. Can cells bind them by means of these two integrins? So I'm gonna give you one examples for this specific collective migration. This is the research paper that I published a few months ago, and they changed integrin subtypes at nanoscale ligand presentation, and they found that by changing this peptides or ligands, cells affect differently. And finally, they found that by having this situation inside the body, we will see that it's not a good way, for example, to have chemotherapy or other treatment for the cancers, because we have to care about the mechanism involved between the cells. Here they have a cancer cells, and they try to have these two type of integrin, which I said earlier, alpha beta 3 and alpha 5 beta 1. And then they have a nano pattern surface, which have a different type of integrin on the different spaces. Then they try to have a drug plus to these parameters, and they studied morphology, migration, and survival rate of cancer cells. If you remember, I said that the RGD domains in the fibrinogen, and cell could bind on the RGD domain by means of these two integrin family. Actually, there are some treatment which block the RGD motif, and doctors are using for as a chemotherapeutic drugs, but unfortunately, they are not sufficient, because they don't care about the spacing which exists in the tumor microenvironment. It's okay for the normal microenvironment, but for the tumor microenvironment, the deposition of the ECM is really messy, and we have a different peptide in the different orientation. So they did some analyzing in this study. At first, they seed cells on the fibrinogen. On the fibrinogen, actually, we have both peptides and domain can cell bind on it. Here you see that cells bind on the fibrinogen, and they stain with actin, paxillin, and nucleus. The paxillin is the adaptor protein which involves for focal adhesion formation. Then they use two different drugs, and they showed that on the fibrinogen, by having this drug, cells has a larger area. And then they use peptodomin. They use the different type of inhibitor that can bind different peptides in the different study, and they showed that by blocking alpha beta 3, cell just can bind on the fibrinogen by means of alpha 5 beta 1. And here you see the difference between two samples. And then also they block alpha 5 beta 1, and then they show that again we can see the differences in shape of the cells, in the focal adhesion formation, and this is the quantitative analysis that shows the focal adhesion area, cell area, and the other stuff that they showed on the normal situation on the fibrinogen coated surface. Later, they tried to have these two different peptides, which immobilize on the surface as the different sizes, and we have 35, 50, and 70 nanometer distances between each peptide. They use Bella copolymer micellar nanolithography, which can give us to have a pattern, a nice pattern to immobilize our specific peptides or proteins. And finally, they could have this pattern to study the cell behavior on the different peptides. So here you see that by having a larger number distance between the peptides, cell has a lower, lower surface area. And then they showed by adding different drugs, cells behave differently in terms of survival rate, in terms of surface area and cell area, focal adhesion area. And they proved just that by changing the space between the peptide, we could say that cell sense differently and cell start to produce different signaling pathway. And finally, we could say that, for example, in this situation, we can apply a lower dosage of this specific drug or higher dosage. Normally, doctor use randomly the different drugs with the different dosage. But in the reality, if we want to go through the cells, and if we want to go and reduce the side effects of each drugs, it's better to have a model system can mimic the actual situation in the tumor microenvironment or also the normal situation. They also did the migration and live imaging, and here they showed that by having a higher space between the peptide, and by having, for example, the second drug cell moving and migrated slowly rather than without the drug. And they showed, for example, I would say that by doing this experiment, they wanted to show the invasion rate of the cancer cells. And they wanted to prove that if the matrix of the tumor microenvironment change in terms of space between the peptides, Integrin sense that domains, and finally we have a different signaling pathway. And it can be one reason that in some tumor we have a very fast metastasis, and the other area just we have a solid tumor can stick on that area. So actually it's really complicated fields which people are trying to understand the signaling pathway at the single cells, and they are modifying and manipulating the structure, and also they are playing with the geometry of the structure and the surface, maybe sometimes without any proteins. And here, for example, they are trying to have a different peptides just to show the different space and distance between each protein and peptides can affect cell behavior. Here they show the survival rate of the cancer cells on the fibronectin, and they show also on the confocal image here. I would say that by using this drug and by engagement of alpha V beta 3, cell can survive in the higher rate. In the opposite, by using the other drug, it has been shown that again we have the higher rate, but the total survival rate is lower than the first try. The interesting result would be for this part, because they showed that for the lower distances between the peptide, we have the lower survival rate of the cancer cells. But when they have a higher distance between the peptide, cell can survive in the higher rate. So it would say that the distance and the involvement of different peptide is the matter for the cancer cell proliferation, the cancer cell invasion, and migration. They did a control on the vintronectin, is another protein which exists in the ECM. And in the vintronectin, just we have alpha V beta 3 integrin. And they showed that by having this type of thing, we could see focal adhesion complex. But if we block this integrin, it's obvious that we can have the focal adhesion, I mean the mature focal adhesion in the cells. And they use another blocker just to block the other type of the integrin. And again, they showed that cell can produce focal adhesion because the vintronectin has alpha beta 3 integrin. Peptide for the alpha beta 3 integrin binding. Actually, the conclusion for this part of the experiment, I mean the research paper, is that they showed the cellular properties of the cancer cells in terms of morphology, focal adhesion formation, or migration is linked to the both integrin subtype and their nanospacing. And if you want to go to the details, you can see the data, because I don't want to go to the detail for this presentation, but later for the other papers as the previous one, maybe we can go to the detail, and also for the material part and this stuff. So today just the concept for the integrin as a biosensor and how cell can sense the microenvironments around the cells or in the tumor microenvironment. Also, they showed that chemotherapeutic drug sensitivity is highly dependent on the both parameter. I mean, with the smaller ligand spacing, we have the lower survival rates. And they showed that by altering also the nanoscale ligand spacing, they are able to increase the cancer cell chemo survival rates is the correlate with the previous finding. And they found that ligand type, a specific pattern of the drug sensitivity with enhanced chemo survival will engage two different integrin on the fibronectin, but by changing the space, they found that cell can die very fast and can be survived on the smaller space. So that's it for the today, and I'm ready to answer some question. Yeah.